Hello, everyone. My name is Yogesh Goyal, and I'm a member of NITMB. It is my pleasure to welcome Jessica Lee from Fred Hutch to NITMB to give a seminar today. So Jessica did her undergraduate degree at Tsinghua University and then moved on to do a PhD in statistics at UC Berkeley, and then started her own group soon after at UCLA, where she spent 12 years building really beautiful work on a range of problems in biological and statistical research areas. And our lab has obviously benefited quite a bit from her methods and approaches in single cell analysis. And recently, Jessica moved from UCLA to Fred Hutch to head their biostatistics program 2 months back. Yeah. And I first met Jessica at a conference, and one of the things I really liked about the meeting was she was probably the favorite of all the students, because instead of hanging out with other faculty, she was spending time with students, which was very nice to see. So she's a big promoter of early career students and faculty, and a lot of her work has been very inspiring. She has obviously also won a lot of awards. She was a Guggenheim Fellow, got an Overton Prize from ICSB, and several others, including Radcliffe Award, Fellowship, and many others. So without further ado, it's my pleasure to welcome you at Northwestern and UChicago's Institute, NSF-Simons Institute, and please take it away.
Thank you. Well, thank you, Dash, for inviting me. It's such a pleasure to visit this new institute and the nice location here. It has such beautiful views. So it's my pleasure also to visit from my new position. So actually, I want to clarify that the title I'm presenting today is slightly different from the ad we posted. The reason is that I wanted to be more engaging for our broader audience to talk about some topics I think we can think together, not just a few technical things, but I want to be broader. And this is actually a talk I previously designed to communicate with my new colleagues at Fred Hutch about some of the topics we can think along, especially does statistics still matter in the AI era? I think that is a question many people wondered about. And also data. How do we actually balance the traditional way of statistical thinking and now the big data? So I'll say that a very big difference I noticed, especially for my research in single cell or spatial transcriptomics compared to traditional statistics, where like you do clinical trials or you do observational studies from population data, is that in omics or modern assays, we often generate data to create hypotheses, not just to test them. So before we actually see the data, we often don't have hypotheses in the mind, but we generate data just because we have those advanced technologies that can give us new information. On the other hand, if you think about classical statistical methods or frameworks, they actually assume that you already have a pre-specified hypothesis, and then you want to use your data to confirm or to reject that hypothesis. So it's a different purpose. And statistics in that sense, in the classical sense, is one of the last steps to give you the conclusion. While here, we are using statistics or computation for exploration. So because of this misalignment, I've seen several issues in current data analysis, which I will talk about later, including double dipping. You use the same data again and again, resulting in like confirmation bias. It could be called, some people in statistics call that post-selection bias. It means that you use the data to select variables or features to look at, but then you use the same data to evaluate those features, right? So because of those issues, you actually cannot easily meet the criteria you hope to satisfy, like type 1 error or FDR, that are often not well controlled. And also these criteria, if you think about them, they're very conservative. They're trying to prevent you from making mistakes. And that is also different from the purpose of exploration. So I want to further say that exploration and confirmation are very different, because for confirmatory studies, like clinical trials is a very, very important example. You have to design the study early on, and then you have to fix your analysis plan. And you want to avoid making false positive mistakes, because if the decision is FDA, whether you can pass the drug or not, you have to be very conservative. The exploratory analysis is different because you are having the data first, and then you can go through data in iterative ways, or you can actually change your analysis pipeline by looking at the data. And the goal is more like signal harvesting or idea generation, rather than having something as the concrete or rigorous result. So because of this, I feel like as a statistician, since we have so many tools for confirmation or confirmatory studies, we are kind of lacking behind in exploratory studies where people try many, many things, but statistics does not seem to be that important. So how can we do? How can we generate or develop new tools for these new questions, not just using the traditional tools that were developed for the confirmatory studies? So that was my big question. So to give you a more concrete example, I would use single cell and spatial omics data we have worked on as an example to illustrate this double dipping. So the problem I'll talk about is called post-clustering inference. So what does it mean? So I'll just first introduce to our audience about how single-cell RNA-seq data is typically generated and analyzed. So essentially, the technology excited many people because now you can measure RNA-seq or gene expression at the genome-wide level. People call that transcriptome in individual cells. So previously, you can only do that for a bunch of cells together. You cannot have the individual cell resolution, but this new technology allows you to do that. So you will have those separation steps of individual cells, so you can barcode or label each cell, and then you capture the RNA, and then you do sequencing. And based on the cell barcode, you can just obtain the expression profile of each cell. This is the data you get. But the data does not tell you the cell's cell type information, because when you do the experiment, the cells in the tissue were just measured simultaneously, and you only have their RNAs, but you don't know what cell type they belong to, like T cells or B cells or blood cells, right? So in this case, we have to rely on some downstream analysis on this data to cluster cells and to give each cluster a cell type label, as a way we call this cell type annotation. Because after you do that, then you can do the analysis at the cell type level, and such results can be transferable to, say, new studies, new patients. Because we know that RNA-seq is a disruptive technology. Every cell, once measured, is gone. You cannot measure the same cell again. But you hope that the information you obtain at the cell type level can be generalizable. So double dipping actually happens in this clustering and then the marker gene identification step. So you're using the same count matrix after pre-processing. Every cell has a vector of genes, and every gene has a count, meaning how many RNA molecules do you capture for this gene in this cell. Based on this matrix, we perform cell clustering in the hope that each cluster represents one cell type. And then we use this differential expression or DE analysis to identify the highly expressed genes in each cluster. And then matching those genes to our knowledge of cell type markers, then we give each cluster a label. But you can see that from this Nature paper, in which I actually turned out to collaborate or consult the second last author, Nicolas, who is now a PI in France, in Paris. So I came across this paper, and I noticed that this is actually very typical in many single cell papers, in that people try to use this T-SNE or UMAP, which are both two-dimensional visualization tools, to project the cells to such a plot. So every dot here represents a cell, and you have those many small cell clusters, and you hope that they represent cell types. You give each cluster a label. But if you look at this plot closely, you will see that many clusters are just numbered without labels. And essentially, they said that 61 of these clusters are labeled based on their menu annotation. But for the remaining, they were just left out there as clusters. So you can see that clustering it's a self and unsupervised approach, since you don't know how many clusters are there, and every clustering method have its own objective function based on their own definition of clusters. So it's very common to have this uncertain clustering results, even though some of the clusters you may be confident about what cell types they correspond to, but for many others you don't. And this is another screenshot from this paper. So if you look at the analysis steps, you can see that this is by data generation for data generation. But since the data was generated in batches, there's another step called batch effect removal. How can we integrate the data together? And that's also part of the data analysis. And then on their integrated data, they do clustering, and then they identify marker genes, and then they annotate the clusters. So for our work, which I will present later, we didn't actually consider integration step yet, but just the clustering and the DE or differential expression analysis together created the double dipping problem. For spatial transcriptomics, which was a newer technology, newer than single cell, you get even more information. So there are 3 types of technologies based on different chemistry for measuring gene expression on a tissue slice. But I want to say that compared to single cell data, the additional information you gained is the location of those cells in the tissue slice. So it's like a 2D spatial information, and you can see which cells are closer to each other, right? The proximity, neighboring information besides gene expression. So people were excited because you get more information, but still you don't know the region of these cells or like the tissue structure. So you don't have that information from the data alone. So nowadays people may have the accompanying, say, histology images or H&E images that can give you additional information about tissue structure. But just based on the spatial transcriptomics data alone, this is what you get. So this one figure corresponds to one gene. So you are just plotting the gene's expression levels at these spots, and the second gene and the third gene. And you want to gain some information or generate new hypotheses from this data. All right. So double dipping here means that if you use the data to define so-called spatial clusters, and there are multiple methods for doing so. People's hope is that each spatial cluster corresponds to one meaningful region, like a functional tissue region or some meaningful substructure, right? You cannot easily see from histology, but hopefully you can identify from this new data. But with those clusters defined, so here the difference is that compared to single cell data where the clustering is only based on cells similarity in gene expression, here you also want to respect the spatial proximity of the spots so that nearby spots are connected into one cluster. But despite that, we actually have the differential expression done to identify highly expressed genes of each spatial cluster, and the same double dipping issue because you use the same data to find clusters and then to find genes. So the question I want to ask is, how much can we trust those clustering defined cell types or spatial domains, especially if you want your conclusions to be generalizable or transferable to new data sets? How can you make sure that you are not having a clustering bias or like over clustering or under clustering, these kind of issues? So in the literature of statistics, this is a well-studied problem for 20 years, the existence of clusters or how many clusters are there. So there are multiple papers trying to determine the number of clusters, but they typically require some distribution assumptions about the data. And also another idea is to say, if you have 2 clusters that seem to be close to each other, seem to be ambiguous, you don't know whether they should be combined or not, there are some statistical tests that were developed to address this issue. However, despite those 2 lines of literature, there is a gap between these methodologies and the practice, because biologists, especially people who do single cell data analysis, they still perform the differential expression test to find marker genes because they want to give markers to clusters. They will not be satisfied with the conclusion just as, okay, the clusters should be merged. right? They want to know, okay, if I keep the two clusters separate, even if their separation may not be so clear, what are the reliable markers? And also, determining the number of clusters, this question is hard to give us a unified answer because we know cell type definition can be coarse or fine, right? You can have coarse major cell types and you can have refined more subcell types. So where do we stop? I think these are all practical challenges. I also saw that some statisticians try to avoid the double dipping issue by proposing a one-step approach. That is, you have one model, you just do model-based clustering, and then you use the model to look for the DE genes. Then you avoid double dipping because it's based on fitting one model. But typically, this will require stronger assumptions, like Gaussian assumption for data. And if not, if you go with negative binomial count distribution assumptions, the model fitting can be computationally intensive. And more importantly, for practitioners, it's harder for them to interpret the results because the step-by-step approach, even though conceptually not good, right, double dipping, but people can look at the clusters, look at the DE genes. It's easier for them to go back to check the whole pipeline. Yes.
So I think I'm just confused how the joint model-based clustering avoids the double dipping. Are you using different data?
No, it's the same data, but it's like one model fitting. As long as you fit a model, the model will itself tells you the clusters and also the marker genes. So you're not doing two operations on the same data. It's like one operation. So because you are fitting one model. So the model parameters will implicitly or explicitly tell you who are the marker genes and who are the clusters. That's why it's called one-step approach. Yeah. Yeah, but it's building. So basically, if you use the one-step approach, you don't have the choice to say, okay, I prefer Leuven clustering versus Leiden clustering versus k-mean. So you don't have the choice of different clustering algorithms, and you don't have the choice of different differential expression tests. It's all building into one model. Yeah. All right, so I will further clarify why double dipping is an issue with some simple cartoon examples. So coming back to the data format, cell by gene, 8 cells, 3 genes. If you first want to find 2 clusters, then it will be based on the 3 genes expression levels, right? So you can divide your cells into 2 clusters. And then if you do the DE test, you have to trust the division. So basically, the 2 clusters are treated as 2 groups or 2 samples, and then you just do, for example, 2 sample t-test to get a p-value for each of the 3 genes. And then if you want to set a threshold on the p-value, for example, to control the FDR, false discovery rate, then you find the genes with p-values below the threshold as your marker genes for either cluster. That is the procedure. But why is this problematic? So if you think about this, I'm talking about the case where there are indeed 2 cell types, but if your clustering is not perfect, in other words, the 2 clusters do not perfectly match the 2 cell types, and if gene 1 is a gene that just got involved in the clustering, so its expression difference drives the division of the 2 clusters, then naturally gene 1 would be defined as a DE gene because of its difference between the 2 clusters, have a small p-value. But going back to the true cell types, gene 1 may be a non-DE gene. So in this case, you can see clustering itself relies on the gene expression. Then the genes involved in clustering will naturally be confirmed as DE genes. But this is a confirmation bias. So that's why we have this double dipping problem. So how do we address this issue? I would say that it's an issue noticed by multiple statisticians. And there are 2 papers from the statisticians Daniela Witten's group addressing this issue using a framework they called selective inference. So I will further say this a little bit without too much technical jargon, but hopefully to explain the idea. But before I do that, I want to say that it has requirements on how you do the DE tests. So it has to be depending on specific clustering algorithms you use. Like they have a paper for hierarchical clustering. They have a paper for k-means clustering. So if you change your clustering algorithm, you need to change how you do the tests. And for single cell data, the commonly used clustering algorithm is actually graph-based. So the idea is that instead of working with cells as vectors of genes in the Euclidean space, you actually first connect cells into a graph, a similarity graph. Then you perform clustering on the graph. So this can actually allow that this cell-cell relationship to be not non-Euclidean, because you're working on the graph, kind of like what people call manifold learning, that kind of idea, instead of just restricting yourself to the Euclidean distance. Anyway, so therefore, the commonly used clustering algorithm cannot be covered, or at least is not yet covered by the two papers. And furthermore, I want to say, what is selective inference? So this line of literature started 12 years ago, and they were actually designed for a different setting, which is prediction problem. So suppose that you have a high dimensional prediction set of predictors, like many features you want to use to predict an outcome. Then how can you know which of those features are important? It's like a covariable selection problem. And lasso was one of the earliest and most commonly used approach in a linear model setting to select the variables or predictors. But if you want to perform statistical tests on the selected features to know what are their coefficients, whether their coefficients, which means their effect sizes, are 0 or not, you have this double dipping problem, because you only have one data set and you already use the data to select those variables. And then if you use the same data to test those variables' coefficients, you have double dipping. So selective inference was actually created to address this problem. How do you use one data set in a valid way so that your second step testing is valid? And maybe I want to step back a little bit to say why we need two steps here. The reason is if it's high dimensional, like you have more variables than your number of observations or your sample size, then the traditional ways of testing coefficients in a linear model would fail. So you cannot use them. So you have to reduce the problem to a lower dimensional problem before you do the testing, but then you have double dipping. So this whole idea of selective inference was for that. But in these two works, they use the selective inference idea, but in this case, the difference is that you are not selecting variables, you are creating new variables by clustering. Because before you don't have the cell type variable, and now you are doing clustering, and you're treating the clusters as the cell types and for your testing. So this is more like a different problem, in my opinion, because one is creating new variables, one is selecting existing variables. But regardless of that, I want to say that selective inference has several key properties I think is very important to know. First of all, what they do, inference, so inference is a jargon statisticians often use, but what actually means is that you are trying to guess parameter values of your population or of your model. And you also want to account for that by this guessing from your data, estimation from your data, there is uncertainty. And the uncertainty can come from multiple reasons. But in the frequentist paradigm, the uncertainty comes from sampling. So you think that my data is just one of the many possible samples I could get. If I get a different sample, then my parameter estimate will be different, right? How much uncertainty is there? Yes.
I'm just curious if you have a comment about why graph-based clustering seems to be so prevalent in single-cell data, because it's not like the data is initially graphical.
No. Very good question. I think that's something I've been wondering for a long time, especially I think in many of those bioinformatics use of networks or graphs, we don't have graph as observed. We have data as matrix or vectors, and then the graph conversion was actually an intermediate step. Why would you do that? And I think my short answer, or more like intuition for single cell data, is because, first of all, your data, every cell is measured as a high dimensional vector of genes, but maybe we don't need so many dimensions. So essentially, maybe the cells are lying in a lower dimensional space, right? And we know that Euclidean distance does not work well. It's not informative in such a high dimensional space. So that's why if you directly apply, say, k-means clustering, which is grouping cells by Euclidean distance in the original space, it does not work that well. I think that's the reason. But On the other hand, what you could do is that people in the field, they typically transform the data using some log-like transformation to make each gene's values across cells more like Gaussian distributed. Then they do PCA, principal component analysis on the data to reduce the dimensions from say 1,000 genes to maybe 20 or 50 PCs, principal components. Then in that space, they think maybe Euclidean distance makes more sense. It's more informative. But then you have another decision. Should I just do say k-means clustering in the Euclidean this space of the PCs, a PC space, or should I further do graph construction? They did the latter. So right now what they're doing is that the cell-cell neighbors or the cell-cell similarity graph is based on Euclidean distance in the PC space. And then they perform clustering on the graph. And for that step, I think the reason they go with this is more from empirical observations to see, okay, that at least the clusters we got seem to agree better with the cell types we know. For the data sets where cell types are like the blood cell data, PBMC, people know, okay, the cell types are relatively well understood. That's why. But I guess the fundamental reason, it could be the reason why the people for doing manifold learning. So I think they don't think the Euclidean distance is that meaningful, but the local distances like the geodesic distance along a manifold may be more reasonable.
So I actually have conjectured myself that it's because the graph-based methods were designed for very, very large data sets and they've been optimized for speed.
That's another thing. Yeah, it's fast. Yeah, even though, that's a very good point. Even though the popular package for single-cell analysis use two graph-based clustering, one's called Leuven clustering, the other called Leiden clustering. They are both heuristic algorithms, right? But the speed wins. Yeah. Thank you for the comment. All right, so coming back to the selective inference, I want to say that here the inference is actually conditional. So in other words, when they talk about type 1 error control for the differential expression or the testing step, it's all conditional on the previous step. Like you are selecting those variables or you created those clusters. So you're conditional on that step and then talk about the distribution of your statistic in the second step. So because of the conditional thing, they actually corrects for double dipping. And also existing work requires extensive derivations for getting the type 1 error control or the null distribution, especially method specific. So in other words, you want to say if I use k-means clustering or if you use hierarchical clustering, I need separate derivations. And in many cases, they even hope to achieve the so-called finite sample guarantee, which means that regardless of your sample size being small, then you can have type 1 error still controlled. So it's like a theoretical criteria they want to achieve. But there's a price to pay. The price to pay is twofold. First of all, like I said, you may have complicated derivations specific to each method, each pipeline. And also, you are actually, if you don't do that, it could be a computationally complex or computationally intensive problem. So that's why I think conditional inference, even though it is something people do in selective inference, but it is not the universal way of thinking about the problem. One reason is if you think about conditional, it means that we trust our previous step. Then we'll only look at the second step. But in many cases, like clustering, that may not hold true because your clustering may not be good. If it's not good, why would you want to be conditional on that? So I had those questions myself. And also I want to further acknowledge that for selective inference, besides those method-specific derivations, recently there was a very interesting proposal I should mention here. It's called data fission or data thinning. So the idea is to create two sets of data from our original data by splitting each data point into two. And this is the data fission paper screenshot. So the idea is that my X is the original data point, say a gene's expression value in a cell. So one value, one number. And the data fission works by randomly sampling a Gaussian noise, Z, and you add the noise or you subtract the noise from X. and then you create fx and gx. So doing so, every x becomes 2 points, fx and gx, and you can use fx to do your step 1, selection or clustering. You can use gx to do your step 2, the inference or testing. But there are some requirements. So in the general sense, you need to know the conditional distribution of gx given fx. Then you use the conditional distribution for the second step, while fx data is used for the first step. So this is a data fission idea. And you can see this is the original data fission paper. And the Daniela Witten's group, which develops selective inference work, they also had this idea, but it's called data thinning or count splitting. So like these words actually refer to the same thing. But the difference here is that for data thinning and count split from the second group, or data fission from the first group, 2 different research groups, data thinning actually has a stronger assumption. So going back here, in data thinning, they're actually assuming fx and gx to be independent. If you have that independence, then you don't need a conditional distribution. You just need the distribution of gx alone, and that should be good enough. But the independence is not guaranteed for sure. You need to have stronger assumptions on the distribution of x so that you can achieve the independence. So my postdoc Chang Hu and my other students and I wrote this comment on this data fission paper because I want to say that data fission was developed for that variable selection problem in supervised learning. It's not a clustering problem we work on. So that's why we discuss how this might or might not work in our post-clustering inference, which is slightly different because our first step is clustering, not variable selection. So we basically went back to check the assumptions, the approach data fission needs to work. So like I said earlier, you need to have one of the 2 properties to hold before you can do anything. First is that fx and gx are independent with known distributions, or fx has known distribution and gx given fx has a known conditional distribution. So if you have one of the 2 properties, then you can use the data fission. But in our case, the complication comes in that, so we are interested in a latent cell type variable we don't observe. So every cell 1 to n has a type. We don't know the type. So we are using clustering to get z hat, which is the clusters, right? And our hope is that z hat is equal to z in the ideal case, perfect clustering. But this doesn't hold for sure. And when this does not hold, we have our problem. So your later inference will be based on z hat instead of z. And if we use the data fission, then z hat is essentially given fx. So z hat should be constructed based on fx. And then we can call this clustering algorithm C. So C applied to fx give a z hat. But later, if you want to use gx, given z hat to do the testing, then you still need to derive this conditional distribution, which is not straightforward what to do. Especially the clustering algorithm can be anything, right? It may not be something you can have a mathematical form for. So that's why we're just arguing that the second step of using the conditional distribution, and in the paper data fission called debiasing, is hard to achieve. While in the data thinning paper or count split paper, 2 names for the same thing, they didn't need to do that because they actually assume a stronger assumption. That is, fx and gx are independent, and that independence has to rely on another assumption on genes being independent. So this is assumption we had question about because we know that in biology, genes are not independent. Otherwise, we wouldn't have like gene regulation, all sorts of things. But without gene independence, we cannot obtain independence of fx and gx. So we need to do the debiasing as in data fission paper, but we cannot do that. So that's why we got into this result that it's an unsolved problem using data fission to solve this clustering-based inference problem. So I'll summarize what I said into 2 challenges. First one, in unsupervised learning, because of the creation of new variables, we are in a much bigger challenge than selecting variables. Second, selective inference as a framework is conditional on the variable selection, or in this case, variable creation, but which may not be reasonable since we may not feel the first step as satisfactory. So especially coming back to the Nature paper I mentioned earlier, right? There are many clusters that the original authors said that they are not confident. So in this case, do you want to be conditional on these clusters? I don't think so. All right, so I want to first give you this problem out here. Clustering followed by testing, it is a challenge, even though it's such a common practice, but we don't have a very principled statistical approach yet. So, but we try to work on that problem, which I'll briefly say later. Another issue I noticed in the field about especially using statistics in genomics, I call it the wild, is that we have this tension. We have biologists who want to actually find discoveries from the data, and they may actually use over-tuning of the pipelines, right, to find something. But this could be vulnerable to technical artifacts and what they believe in the data, like the prior hypothesis. On the other hand, if we go with rigorous statistics, we may have a procedure that gives you near zero discoveries. You can say, okay, given the noise of the data, I cannot find anything. And that is something scientists would not like. So I'll give you one example of showing that over-tuning biologists versus conservative statisticians, which was a very recent debate. And this was a Nature paper published last year from a famous neuroscience lab at Stanford, right? They generated spatial transcriptomics data, and their major claim is that they found certain genes related to long-term memory in certain cell types. All right, and earlier this year, there was this Matters Arising paper published in Nature from 2 statisticians who claimed that the study found only false positives. Okay, let's revisit their debate and see what happened. This is from the Matters Arising paper, arguing that the original Nature paper didn't perform what we called multiple testing correction on their p-values. So they just applied p-value threshold 0.05, the nominal threshold, and try to find those genes in red as the so-called differential expressed genes between, and it's actually a cell type they are interested in of mouse data, and they look at 2 conditions. One has relationship with long-term memory, one not. So that's why they hope that the differential expressed genes can be related to long-term memory. That's the whole idea. But if you look at these genes after you do the FDR correction or multiple testing correction, none of them could pass the 0.05 threshold, meaning that you have 0 discoveries, right? Another point is that the original paper used every cell as a measurement unit. So the testing was performed across cells. But the authors of the second paper argued that they should treat each animal as a unit and aggregate cells within each animal, because the animals are more like independent units, while if you look at the cells, then the cells within each animal will tend to be more similar, right? So you cannot assume all the cells to be independent. That's a second point to point out. But let's just argue that overall, the second paper think the original paper should not have any reliable discoveries. But as the format for Matters Arising, the original authors needed to reply. So there's a reply from the original authors, and the reply contained multiple sentences that made statisticians concerned. And I was actually pointed out to this debate because people said, okay, you should take a look. And there are many interesting words. So basically, the first paper, original authors argued that the Matters Arising statisticians took a narrow extreme position and also overly stringent, and said that such corrections depend on specific context. So they're trying to fight back by saying that we do not always need multiple testing correction. Anyway, so they're challenging that the conservative perspective of not making too many false positive discoveries. And they said that they want to take a more relaxed approach. And they also argue that it's less of a concern for their discoveries because they use independent experiments to confirm their discoveries. So basically, I want to say that if you look at the paper carefully, the analysis criticized by the second paper was for single cell data, and that same Nature paper performed spatial transcriptomics data. So they were saying that the spatial transcriptomics data confirms single cell data. But there's another concern I had for that, because for both data sets, the results are computational. So you're using computational results to confirm computational results. But we know that computational results can be subject to the pipeline choice. Anyway, so they said that, okay, if we want to follow your suggestion of doing multiple testing correction, let's do it ourselves. So instead of working with the original 3,000 genes, they actually, zoom in, or they screen down to 56 genes. And they only analyze those genes and then do multiple testing correction. And they argue that, okay, now we have discoveries. But the problem here is that you already use your data to get down to 56 genes. So it's again double dipping. So, right, because you're using your judgment to say, okay, I think these 56 genes are the more reliable ones, then I don't have so many tests to correct for. So that's why I can find discoveries. But this made us very interested. So my student in Chenxin, she looked into the data. I suggest her to look at data. And let's show the data to you first, then let you see what it looks like. So this is the original data. We look at the genes reported in the Nature paper, but couldn't be found as significant in the Matters Arising paper. So there are the two conditions of each gene. So you can see that if we just do eyeballing of the data, we could see some differences in the two distributions, right? One from each condition. But if you look closely, it is not surprising to us that if you do differential expression, you wouldn't have many discoveries, because the mean difference does not seem to be so different. However, what seems to be more different is the variance, right? You will see that a gene has a much larger variance in one condition than the other. So that's why I suggest that my students to check the differential variability. Maybe I should go here to see whether there are meaningful variance differences. And let me just go further. And indeed, which is very interesting to us, is that if we do the differential variability analysis in a rigorous way, in our opinion, we can still recover a portion of the originally reported genes, especially the two most important ones here. So I think here, and also I didn't show the results in the previous slides, which basically we confirm the second Matters Arising papers analysis. Using our independent analysis, we also say that, okay, if you just go with differential expression, we don't have discoveries. But we change our angle to look at differential variability, then we have some meaningful discoveries, which can partially confirm the original paper. So in my opinion, we have done the reconciling view of the two the two sides of the debates. Because if you think about the Nature paper Matters Arising, the two sides didn't reach an agreement, right? One side says no discoveries. The other side, the original author said, no, you are too stringent, you are too narrow-minded, then we have discoveries. So they stop there. But we're basically saying that two sides have potentially some points, but I think the key thing we want to argue for is that there's a mismatch. So if the data doesn't contain strong differential expression signals, you shouldn't forge your analysis to find DE genes. But you should think about, maybe I should look at different angle or different perspective of my data and see what I'm looking for.
Jessica, but does the variability in the DE genes, is that supportive of the claim they make biologically, or the biological claim was it's more or less? Because that's a different claim then, right?
Exactly. But actually, I think they are just trying to find some potentially interesting genes associated with long-term memory. Yeah. And here I will say that if you look at the one, so this is like the one without, let me see. I think if I remember correctly, so the wider one, the blue one, is the no memory part, and this is the memory one. So essentially, you can see if the gene expression has some distribution change, maybe like shrinking towards like a narrower range, it may mean something, right? Because this is single cell data, and we know that every cell is not the same. Yeah, I think the heterogeneity we can see in single cell data, but not in bulk data, may also be a valuable point. Right? So like, yeah, that's what I think. Yeah. All right. So the take-home message, I'll be quick given the time constraint, is that We hope to find a reconciliation by looking at both sides and see what we can offer. But our recommendation is still we should avoid double dipping, and we should look more than just the common differential expression, right? That is something people all do, but in your data, maybe the shift or the change is not in the mean value, but it could be in other aspects. And another thing I didn't talk about further is that we tried a lot of permutation to create a null case. And we also tried perturbation of the data or subsampling of the data to create a stability analysis to confirm our results. And which is something I think is very important, because if I go back to the DE part, which I skipped here, we want to argue that if you do the permutation, then you can see that for many, many permuted data, DE-seq2, which is consistent with our previous paper, it can find many, many DE genes. So that's why if you use DE-seq2 and see DE genes, you have to be cautious. If you can also see many discoveries from permuted data. And we also tried other things. But the Nullstrap, which I will say in the last words, our approach is a way to correct for those FDR inflation. And on this data set, with the application of Nullstrap, we didn't find anything. All right, so moving on to my last part. So I would say that for statisticians, we need to think about more ways of providing useful criteria for discovery-centered research, right? How can we still help people do discovery, not being so conservative, but we can offer more principles rather than just ad hocs. So in particular, I feel like we need to prioritize power, discovery power. The opposite of that is type 2 error, more over just type 1 error. Because maybe for early stage discoveries, you would rather have false discoveries, but you want to retain your signals. So that is a very different perspective compared to the confirmatory analysis. Okay, so I want to say that coming back to that variable selection problem, right? I mentioned that earlier, supervised learning. So in statistics, there's one approach called knockoffs, which has been very strongly advocated by the developers in the Stanford School of Statistics. They are like the major players in statistics field. And the idea is very interesting. So the idea is to create some synthetic contrast or controls by generating knockoff features, which look like the original features, like a P genes. You generate P fake genes, like knockoff genes, but you want these knockoff genes to have no relationship with the outcome you're predicting. But they have the same correlation structure with some exchangeability property with the original feature. So they're fake enough, and you cannot easily tell them. Then essentially for any models, you can include those P knockoff features together with the original P features, and then you let them compete with each other in the model. Then that can help you identify which of the original P features are truly meaningful. So that is a very interesting idea. But the question that motivated our research is that by adding those fake features, you're making your original data worse. So you're sacrificing your data quality to achieve this FDR control. Because in knockoff literature, FDR control is the only goal, and you are doing that by decreasing the data quality. So we wonder, and that will sacrifice the discovery power, which was what I said in the previous slide. We should focus more on the power. So our idea is to preserve the original data, but we still want to use something synthetic as the null or negative control, but we use that in parallel instead of combining the two data sets into one, because that will decrease the original data quality. And also because we're going for this parallel idea, we don't need to make the synthetic null data to satisfy strong properties with the original data, such as you can swap the variables, and that will make our data generation a much simpler process. So for these reasons, and I also want to say that this is the core idea behind Nullstrap. So the name Nullstrap actually came from Bootstrap, but the Bootstrap idea is to generate data like your original data,
by either resampling or you feed a model to your data and sample from that model. In our idea, Nullstrap, we are trying to sample from a null hypothesis or null model. So the model is modified to reflect our null hypothesis. And we also have some roots in our idea in this classical likelihood ratio test. If you think about it, what you're doing here is that you have a null hypothesis, which is about some constraint of your parameter value. You have a less constrained alternative hypothesis. And you fit your best model under each hypothesis for the same data. So this is by maximizing the likelihood, right? You find the most likely model under each hypothesis. Then you compare the two likelihoods to see whether their difference is meaningful. So that's the idea. And in our approach, instead of working with the likelihoods, we turn each likelihood into a data set. So for the less constrained alternative hypothesis, we just work with the original data. For the constrained null hypothesis, we sample from the fitted model, we get a null data set, and we compare the two data sets to make discoveries. This is our proposal, Nullstrap, and it's actually trying to solve the original variable selection problem in the supervised learning. And we showed that for all the things existing approach can do, like the knockoff approach or data splitting approach, we can also do that. We can do the same variable selection problem. But notably, we have higher power because we preserve the original data, and we are fast because we don't need to have the complication in data generation as in knockoff. And also, we only need to do the data generation for once. Then, because we have many, many variables to compare, and they themselves form a group of comparison. So one generation of null data will be sufficient for the selection. So I'll just quickly show one example of how it applies to. So this is a data set in which you have much higher number of variables, right? This is the number of omics features, and N is the number of pregnant women. And you're trying to use their blood's omics features from three types of omics, metabolomics and something else. Try to see which of the omics features are associated with their time to labor onset. So it's like a survival kind of thing, but it's a pregnancy. So essentially, this data set was previously used by the knockoff and also data splitting. Try to see what variables they can select or feature they can select. And all the three procedures, including Nullstrap, are randomized procedures. So we depend on the random seed. So that's why we run it for multiple times. Here we run it for 70 times and report the number of features we select. So notably, you can see that multiple data splitting tend to select nothing. And I think the reason is because your sample size is already very small. And if you further split it into halves, you get 75, right? So you don't select anything. And Model X knockoff, for most of the times, nothing. Sometimes it selects something. While using our Nullstrap, you can see that we can select variables across different runs. And if we look at the selected variables prediction accuracy in terms of just the R-square, we can do very well. And also to our pleasure, there was an independent benchmark that was actually proposed to benchmark those variable feature selection methods. And this Nature Biotechnology paper had this method called stable. And stable is actually based on knockoff. So it's a multiple runs of knockoff and then ensemble to become stable. So it's a knockoff-based method. And knockoff, another issue with knockoff is that it's very, very slow because of the generation. So in the comparison, we're very happy to see that this independent group reported Nullstrap, though still unpublished as a preprint, had the best accuracy in terms of area under precision recall curve or F1 score for distinguishing true features from null features in their simulations. And also compared to some standard approach like random forest for variable selection or mutual information for variable selection, we're also better. And in terms of computational time, you see that Nullstrap is the fastest, even faster than mutual information, while knockoff, the stable, is actually much slower. So these all give us confidence. And I want to say that the Nullstrap is a generalization from what we did for the single cell double dipping method. So I didn't have time to go through that method, but that was the original motivation. That is setting up some synthetic null data so we can do the inference, unconditional inference for the whole pipeline. We consider what clustering is doing and what testing is doing instead of just being selective on the clusters. So I want to argue back to say that selective inference, even though it's very interesting, but it has challenges in real applications. For example, either the derivation is too complex or you need strong assumptions like in data splitting. So that's why we think setting up synthetic null data and use that in parallel from the beginning is a very general and useful idea. We hope to push to more applications. And Nullstrap is just one generalization we recently proposed. And also, for our audience who know more about like statistical theory, I want to say a little more that we did sacrifice something. It's not free lunch. So compared to knockoff, what we sacrifice is what they called finite sample FDR control. So in other words, our FDR control is asymptotic. We need a sample size to be large enough, and we need a model to fit well to the data. While for knockoff, they try to argue that, okay, we can have FDR control for no matter how small the sample is, but they have their assumptions as well. But I just want to argue that probably that perspective is also a little bit too conservative, because if you want to achieve that, you have to lose power. And the power loss seems to be too significant to me. And also the computational cost is another thing to concern. So I want to say that I didn't have time to talk about the technical thing, but for our audience, if you are interested, this is the preprint, and we have all the details here. And so far for the variable selection problem, we have shown that Nullstrap can work very well for linear models, generalized linear models, including like Poisson negative regression, and also Cox regression models for survival analysis, and also Gaussian graphical models for network inference. So we have shown these applications. So finally, this is my last slide because I want to stop here by saying that I think giving the AI challenge or AI as a useful tool for us, I have been thinking how we can generate what we call the synthetic null data from AI. But the challenge here is that it's not easy, at least to my knowledge, to specify what is a null hypothesis using AI, right? Because right now, I think if we use AI for data generation, we are trying to mimic real data. But how can we inject our null hypothesis, kind of like a constraint into the AI model before we generate data? I think that is a very big open question we can think about. All right, so finally, I think this is something I presented to my colleagues. I would just say that I think practice research has a lot of mismatches, and then also whether our research is on the basic side of biology or trying to be translational or clinical science, we have different needs for evidence thresholds or incentives, right? Maybe for basic biology, we can be more bold, right, in terms of making discoveries and then confirm in the end. While for clinical research, since your decision based on statistics is so important, last step, you have to be conservative. And also black box performance versus mechanistic insight. I think this is also related to NITMB. Many people who study mathematical biology may focus more on mechanistic insights rather than black box prediction, right? And also what statistical habits can travel well to problems, which should we retire, like the FDR control thing, right? We have so many work on that, but which of them are really useful? And this is something I said, discovery operating characteristics. Should we think of what kind of criteria we need? Because I feel like in this AI age, we also need more metrics that can be meaningful for our research, because the problems we work on are not exactly the same as like writing text or generating images in daily life. Yeah, so, and finally, the support of transparent exploration. I think transparency is always something we need to think about when we use black box. Yeah, so that's all. Thank you very much.
Any more questions? I know we have some questions during the talk.
Yes.
I think it's a very interesting concept for the AI generated now data. I think it's a very interesting idea.
Thank you.
The one problem is that very often, sometimes entire scientific domain is a bias. Scientists are biased. Our brain is biased for certain reasons, social reason, whatever reason comes in. I'm just curious. So in that case, AI now data might be also biased because it depends upon
I agree.
put into it.
I agree.
So what's your idea? Is it possible to avoid it, or what's your comment?
I agree. So this is something I also talked to it. Actually, it was the fallback forum. We had this interesting discussion. And I think some doctors and basic scientists have concern about using AI to generate new scientific hypotheses. The reason is that they think we only report positive results in our papers and negative results were left out. But AI's ChatGPT or Gemini, so these AIs were trained on papers, right? So therefore, they will be, how to say, have the bias of follow existing discoveries. They wouldn't have the chance to see the other side, like the missing discoveries, the negative results. So then if you rely on AI too much, you are basically trapped into this conventional way of thinking, existing knowledge, existing discoveries, and it's harder to be disruptive. Because if we think about a lot of the new or important scientific discoveries, they were disruptive. They challenged existing theory. They said something was wrong, right? Then we propose something new. But you will be missing the opportunity if you rely on AI too much. So that's why I really agree with you, Professor Long, that we can think of how we can find the negative side of AI results. So can we actually intentionally look for what AI doesn't give us? And that may be interesting.
But so the take-home message is that even for the AI general thing, we have to be critical.
Exactly. We have to be critical. Yeah. Otherwise, we would just be following the crowd. Yeah. Thank you.
Thank you so much.
Thank you so much, Yogesh. Yeah, thank you.